Welcome back. In the next series of web lectures, we're going to be taking a look at some patterns of inheritance that are different in many ways from what Mendel observed directly. And so they're going to largely follow Mendel's laws, but they're going to do it in a slightly different way. So in the last couple of classes, we have been thinking in depth about how the behavior of chromosomes during meiosis results in the laws that Mendel observed. So specifically, we saw that the law of segregation occurs when the two homologous chromosomes are separated from each other in meiosis 1, when they're pulled over into separate cells and are deposited eventually into individual gametes, that each offspring will only get one of those two alleles. So that is where that happens physically. And then the law of independent assortment occurs when the two homologous chromosomes line up together on the metaphase plate during metaphase 1, and they do this in random order with respect to the paternal and maternal homologs. So they can either line up together, maternal with maternal and paternal with maternal, or they can mix and match, and with many more chromosomes, many more different combinations of maternal and paternal chromosomes are possible. So this is where the law of independent assortment happens. But for a couple of generations after Mendel made these discoveries, chromosomes had not even been discovered. Mendel knew nothing about chromosomes, and that made people very skeptical about this whole business of these genetic factors that Mendel talked about. Nobody really knew what these factors were, and it didn't really make sense in terms of what was known at the time. As people started to observe the behavior of these chromosomes, first during mitosis and then later during meiosis, they began to make the connections between what Mendel was talking about and what they were seeing these chromosomes do. And some of the earliest experiments that really established chromosomes as the units of inheritance were discoveries that were made regarding sex-linked inheritance. So this information can be found in concepts 15.1 and 15.2 in your book. So the first solid evidence associating a specific gene with a specific chromosome came in the early 1900s from the work of a scientist named Thomas Hunt Morgan, one of the very earliest geneticists. His early experiments provided convincing evidence that the chromosomes are in fact the location of Mendel's heritable factors. So what did Morgan use for his experimental species? Morgan chose to study Drosophila melanogaster, which is a common species of fruit fly. So this species has a lot of the characteristics that we talked about previously of a model organism that is very convenient to work with in the laboratory. So they produce many offspring, so you have very large sample sizes from each cross. A generation can be bred every 2 weeks, so a very short generation time. You can get a lot of results in a short period of time. And they have very simple genomes. They have only 4 pairs of chromosomes, so they're very convenient to work with. So Morgan noticed that there were wild type or normal phenotypes that were very common in the fly populations. And actually he painstakingly bred populations of fruit flies generation after generation, looking for some kind of variation away from these wild type features. And eventually he found one, and that was the white eye trait. So traits alternative to the wild type are called mutant phenotypes. They arise by mutation to the wild type gene. So the first mutant Morgan discovered was a fly with white eyes, and it was a male, instead of the wild type red eyes. So let's take a look at how Morgan was able to associate this trait of eye color with a particular chromosome in the fruit fly. So he did one experiment where he mated male flies with white eyes, so the mutant allele, with female flies with red eyes, or the wild type. So as expected in a hybrid cross between two pure breeding lineages, the F1 generation had all red eyes. This established that red was the dominant allele. The F2 generation showed the expected 3-to-1 red to white eye ratio, just exactly like Mendel's traits, but with an important difference. Only males had the white eyes. So all of the females had red eyes, and half the males had red, and half of them had white. This was very unexpected. So Morgan reasoned that the white-eyed mutant allele must be located on the X chromosome to give it that sex-specific expression. Morgan's findings supported the chromosome theory of inheritance because it was known that sex determination occurred due to the combination of these X and Y chromosomes. So let's take a look at what happened in this cross. So here's the experiment. We had females with red eyes crossed with males with white eyes. All of those F1 offspring had red eyes. In the F2 generation, we get the 3-to-1 red to white ratio is expected, but again, all of the females were red-eyed and half the males were red and half were white. So here is what Morgan inferred from that. So if we have the 2 X chromosomes for the female, each of them with a wild-type allele, and then the male, which is XY, the X chromosome has a mutant allele on it, and the Y doesn't have any allele on it at all. The Y doesn't carry that gene, and so there's just the one copy of the mutant allele. So then when we do our Punnett square, this is reduced because the female only has one kind of allele, so there's no reason to repeat those squares below, so we do it in just one row. The wild-type allele from the female can either pair with the X chromosome with the mutant allele from the male, giving females that are heterozygous, and so with red eyes, or that wild-type allele from the female can pair with the Y chromosome, giving males, but this is a wild-type allele, so they are also going to have red eyes. So in the F1, we've got heterozygous females and males with just the single wild-type allele. So now when we do this back cross, crossing these heterozygotes with these males from this cross. Now doing the Punnett square, we've got the heterozygous females being crossed with the male with the wild type and the Y with nothing. So we get homozygous females and heterozygous females, about half and half, homozygous and heterozygous. Half of the males will get the wild type allele from the mother and the Y chromosome, of course, from the father to give you the XY male. The other half will get the mutant allele from the mother and then the Y chromosome from the father. And so these are going to be the white-eyed males in that second generation. And so this is how X-linked inheritance works. So Morgan's discovery of a trait that correlated with the sex of flies was key to the development of the chromosome theory of inheritance. Now we had a definite link between these heritable factors and chromosomes. So let's talk a little bit about sex determination in humans and other animals. So humans and other mammals have 2 types of sex chromosomes, a larger X chromosome and a smaller Y chromosome, a much smaller Y chromosome. A person with 2 X chromosomes develops as a female, while a male develops from a zygote with 1 X and 1 Y chromosome. But there are other patterns of sex determination possible in other animals. So here is the typical pattern for humans. Males have 44 autosomes plus X and Y. Females have 44 autosomes plus X and X. So all of the eggs that a female produces have X chromosomes in them. Of the sperm that male produce, about half have X and half have Y. And then the zygotes, the offspring, will always get an X chromosome from their mother and 50% chance of getting X or Y from the father. But other systems are possible too. So in some insects, there's the XO system where the females have 2 copies of the X chromosome. The males just have 1 copy of an X chromosome but no Y chromosome. So there's 1 fewer chromosome in the males in these species. In birds, there's a different system in which it's the males that have the 2 copies of the same chromosome. So we call this the homogametic sex, where there are 2 copies of the same chromosome, and the females have 2 different chromosomes. We call this the heterogametic sex. So we call this ZW for females and ZZ for males. So as opposed to in mammals, where the females are the homogametic sex and the males are the heterogametic sex. In insects such as bees, we call these hymenopteran insects, they have a system of sex determination called the haplodiploid system. And so this is really interesting. So the females are diploid. They have 32 sets of chromosomes. The males are haploid. They arise from mitotic division of unfertilized eggs. And so they never restore that diploid number of chromosomes. So they have no sex chromosomes at all. The difference between males and females is that the females are diploid and the males are haploid. So There are short segments at the ends of the Y chromosome that are homologous with areas on the X chromosome, and that allows the two to behave like homologs during meiosis so that they can match up and connect together during meiosis in males. There's a gene on the Y chromosome called SRY, sex-determining region on the Y chromosome, and that's responsible primarily for the development of the testes in the embryo. In general, a gene that is located on either sex chromosome is called a sex-linked gene. Now, the genes on the Y chromosome are called Y-linked genes, and there are very few of them. You saw how small that Y chromosome was relative to the X chromosome. There are only about 78 genes coding for about 25 proteins. Genes on the X chromosome are called X-linked genes, and the human X chromosome contains about 1,100 genes. So the X chromosomes have many, many genes for characteristics that are completely unrelated to their sexual identity. So many Y-linked genes are related to sex determination. So as we saw, these X-linked genes follow a very specific pattern of inheritance. For a recessive X-linked trait to be expressed, a female needs 2 copies of the allele. So the female would have to be homozygous, but the male only needs 1 copy of the allele to be expressed, and we call this state being hemizygous, having just 1 copy, but there's no other copy to interact with it in any way. X-linked recessive disorders are much more common in males than in females because of this, because they only need to get 1 copy rather than matching up 2 copies. So if we think about the example of colorblindness from your book, in this figure, X with a lowercase n superscript indicates the recessive colorblindness gene. The uppercase n refers to the normal wild-type gene. So if you have a female homozygote crossed with a male who's affected with colorblindness, if you do the Punnett square, you will have half of the offspring being female, and all of them will be heterozygous carriers of this colorblindness allele, and none of the males will be colorblind, even though their father was colorblind, because they're getting their X chromosome from their mother who had only wild-type normal alleles. So, on the other hand, if we look at a heterozygous female, one of these carriers, mated to a male with the normal copy of the allele, now if we do the Punnett square, what we see is for the females, half of them are homozygous wild type, half of them are heterozygotes, all of them with normal vision, but now half of the males are going to have the normal vision, and half of them will have inherited that recessive mutant allele and will be colorblind. If, on the other hand, you have a mother who's a heterozygote who mates with a colorblind male that has that mutant version of the allele, now you've got a 50-50 chance of the female being a homozygote recessive and being colorblind, and 50% chance of a female offspring being normal and a carrier, 50% chance of a male child being normal, and 50% chance of the male child being colorblind. Here are some of the disorders caused by recessive alleles on the X chromosome in humans. We've talked about color blindness, which is mostly X-linked. Most of the causes of color blindness are due to X-linked recessive mutations. We talked about baldness. It's not on the list from your book, but that is also a recessive allele that is sex-linked. A particular form of muscular dystrophy that's prevalent in men is also an X-linked recessive disorder. And also hemophilia. This is a disease that inhibits normal clotting of blood. So I mentioned in the last web lecture when I was sort of leading into this topic that you can think of these disorders that are found primarily in males. You can think of them as being the fault of their mothers. So let's take a look at why. So if you look at a pedigree for a typical X-linked recessive trait. So in this case, we've got individuals that are affected by the trait in blue. We've got carriers half shaded. And so if you imagine that you're a male and this is you right here, you got your one X chromosome from your mother. So here's your mother, the heterozygote. And of course you got your Y chromosome from your father, but it doesn't really matter because that allele is not carried on the Y chromosome anyway. So she got one X from her mother and one from her father, but her father only had one X chromosome, only one allele. So she had to have gotten that one recessive allele from her father and then a normal one from her mother. So if you're colorblind, it's because you got that allele from your mother who got it from a father who was colorblind. Similar thing for baldness. You inherit your baldness from your mother, as strange as that may sound. So an interesting phenomenon occurs in mammals, in the females. At an early stage of development, one of their two X chromosomes in each cell is randomly inactivated during embryonic development. So what this means is that females get the same gene dosage. They still get the effect of just one of their two X chromosomes, the same as males would. So let me explain a little bit more how this happens. So here we see a fertilized egg, a zygote at the one cell stage, and it's starting to divide. At some point, when it gets to somewhere around the 32 to 64 cell stage, every one of these little cells is going to just randomly turn off one of its X chromosomes. And that's a completely independent, random event in each one of these cells. The inactive X condenses into a structure called a bar body. This condensed version of the chromosome, as many of you will remember from B core 11, is not able to be transcribed because the transcription factors cannot actually physically reach the sites on the DNA to be able to attach and transcribe that DNA. So just that state of being perpetually condensed prevents that gene from being read and transcribed and translated. So what happens is that all of the cells that descend from each of these early developmental cells after mitotic division are going to have the very same X chromosome inactivated. So that's going to be sort of a state that's perpetuated through mitotic divisions. And so what you get is sort of a mosaic patchwork of different X chromosomes being turned on in different cells in the organism. And so this is what that would look like. So let's say for a cat, you've got an allele for orange fur, an allele for black fur. Somewhere down here at this early developmental stage, one of those X chromosomes is randomly shut off in each of these. In this cell, it's the orange fur that's condensed into this bar body. In this one, it's the black fur allele that's inactivated. That results in this color pattern that we see in tortoiseshell cats, this kind of mottled appearance due to the different cells being located at different parts of their body that are expressing these different genes. So this is why tortoiseshell cats are nearly always female, and calico cats are nearly always female too. They have the same phenomenon going on, but they also have a separate gene that causes the white patches that you also see in calico cats. So calico cats and tortoiseshell cats, nearly always female. So if a female is heterozygous for a particular gene located on the X chromosome, she will be a mosaic for that character.